Hello, everybody. So the second set of information, we just did hairs. Now we're going to do fibers. So the fiber section is fairly shorter, okay? It's better to actually do the identification of the fibers than to talk about how to identify the fibers. So it'll be a fairly short presentation. I think this is like 10 slides or something like that. You'll also have access to your book, so you'll be able to reference anything that I talk about. So let's get moving. So in terms of trace evidence and fibers, there are two different types of fibers. So the first one that we're going to talk about are going to be natural fibers. These are the ones that are going to be derived from either animal or plant sources. So some of those examples are going to be mohair, wool, different furs, cashmere, and of course, cotton. The other type of fibers, to distinguish them from natural fibers, are going to be man-made fibers. So these are basically regenerated fibers that are manufactured from natural raw materials. So it's like we took those natural raw materials and refined them and made other types of fabrics. And some of those examples are going to be rayon, acetate, and triacetate. In terms of synthetic fibers, they're going to be produced solely from synthetic chemicals. And some of those are going to be nylon, polyester, and acrylics. Now we're going to get into a little bit of the chemistry behind fibers, not very much, but you need to understand how they're created. So when we talk about polymers, a fiber is a polymer. So a polymer, or as you can also call it a macromolecule, are going to be synthetic fibers that are going to be composed of a number of atoms arranged in repeating units, and they're usually the same repeating unit, and we call those repeating units monomers. So if you take a look at this picture, this is just showing you a link in a chain, and every single one of those links is identified as a monomer. You put 5 of them together, and guess what? Now we have a polymer. Polymer meaning many, mono meaning one or single. So if you take a look down below, here are just some 2 components that are naturally made in nature, and that would be starch and cellulose. They are basically monomers of glucose. The difference between starch and cellulose is that they're just connected at different spots in the glucose chain, which gives it a different structure and a different formula. All right, so let's talk about how we look at fiber evidence. So for fiber evidence, we're going to do a microscopic comparison between the questioned and the standard reference fibers. What we're going to be looking for is going to be color, diameter characteristics, and we are going to use a comparison microscope. Now, we start talking about how to look at fiber evidence. The first thing we're going to look at is morphological features. We're just basically going to look at it with the microscope to see what's going on with the shape. So it's important in comparing the fibers, and these are the 4 things that you're looking for. You want to look for lengthwise striations on the surface of the fiber that you can compare with the suspect fiber or the crime scene fiber. You want to look for the presence of delustering particles that reduce shine. Certain fibers, let's say, shimmer, some don't, so you want to look for those types of things. You want to take a look at the cross-sectional shape of the fiber, so you can do a comparison that way. And then you obviously want to look at the differences in the dyes. So manufacturers, they may manufacture the fiber the same, but their formula of the dye that they use is going to be different depending on the manufacturer. So some of the methods for fiber comparison are going to be using a visible light microspectrophotometer. What that's doing is it's comparing the colors of the fibers through a spectral pattern, meaning that it's looking at all the different colors that are coming through on the fabric. So if you take a look on the right-hand side, this is just showing you a microscopic view. Basically what you're doing is just shining a light through the fiber, and depending on the fiber, it's going to come back or shine back a different color. The other thing is you can always look at the fiber's dye composition, and you can do that through a chromatographic separation. We'll talk more about that when we get into gas chromatography and some drug analysis. And then we'll also talk about a little bit more when we do some ink comparisons with ink chromatography in our documents unit. So essentially the process, you're going to use solvents to separate the dye, and then there's going to be a machine that's going to quote unquote read each color. So the other thing that you can be doing is using an infrared spectrophotometry. So it is fast, it is reliable, and you can identify generic classes of fibers, and also the polarizing microscope will be very helpful. Depending on the class of fiber, each fiber is going to have a different index of refraction, meaning that there's going to be a certain sweet spot for that particular fiber. You shine a light through it, it turns a certain color, and every fiber is going to have a different index of refraction. So you can actually use that value to make a comparison. In terms of collection and preservation, the investigator's task is slightly cumbersome when it comes to fibers, because what you're doing is you might find a sweater somewhere and there may be a couple of fibers on the sweater that you need to collect. But you're going to have like that sweater is going to be the carrier of potential fiber evidence. So you have to look really closely at it, because if you bag the sweater and let's just say miss that fiber evidence, you might not be able to analyze it to figure out what's going on at your crime scene. When we're talking about how to package them, you definitely want to carefully package them in separate paper bags. The investigator is going to use clean forceps. It's going to place it on a small sheet of paper. If you guys remember from the very first unit when we did packaging, it's called the druggist fold. You're going to fold, you're going to label the paper, you're going to place the paper packet inside of another container. So it's obviously, it's like you're double bagging it, right? Because you don't want to lose it. Hair and fiber evidence are notoriously easy to lose. And we never use anything plastic, obviously, because plastic will create static, and static can be a problem when you're trying to get out your fiber or your hair, or you might not find it in your plastic bag. So it's always a little bit more difficult. But in general, remember, we always use paper, not plastic, right? So that's pretty much it for fibers collection. We're going to try and do a fiber comparison activity. Either you guys are going to be watching some videos on some burn patterns of fibers, or we'll figure out some sort of virtual microscopic activity. All right, so if you have any questions, you know where to find me. Bye.